This video will demonstrate the histology of the lower digestive system. So we're going to start off by talking about the duodenum, which is the very first portion of the small intestine right after the stomach. And we'll see the layers of the wall, the mucosa, submucosa, muscularis externa, and serosa. You notice those were the same as the stomach. The mucosa is still going to be simple columnar epithelium, and the surface of all of these cells are going to have microvilli on their apical surface, and microvilli increase surface area for those cells. There are going to be larger finger-like projections into the lumen of the small intestine called villi, and just like the microvilli, these help to increase the surface area of the small intestine. And in the center of each villus, there's going to be a lymphatic structure called a lacteal, and this is where the lipids that are absorbed go into the lacteal, then they go into the bloodstream after that. The submucosa has some intestinal glands. Those are going to make some secretions out onto the surface. And the muscularis externa has two layers, an inner circular and outer longitudinal. And on the outside there will be a serosa. When we move on to the large intestine, there will still be the same layers, the mucosa, submucosa, muscularis externa, and serosa. But really what we're going to be concentrating on are the fact that we will see lots of goblet cells. There are going to have to be several here to ease the movement of feces through the colon because water is being reabsorbed. This process is called compaction, and so we need lots of goblet cells producing mucus. The accessory structures here are going to be the liver and the pancreas. And when we look at the liver, the hepatocytes, or mature liver cells, are going to be arranged in a six-sided figure called a lobule. And at the center of each lobule, you will find the central vein. And the channels that lead to the central vein are going to be called sinusoids. At the corner of each lobule is something called a portal triad, and it's going to consist of a branch of the hepatic artery, the hepatic portal vein, and the bile duct. The hepatic artery is bringing fresh blood from the heart to the liver. The hepatic portal vein is bringing blood from the digestive organs to the liver, and the bile duct will be leaving the liver containing bile. The pancreas is considered a mixed gland because it has both endocrine and exocrine functions. The pancreatic islets are little islands, and they're going to be producing the hormones insulin and glucagon. And then the pancreatic acini produce the digestive enzymes that will be delivered to the small intestine to aid in digesting all of the food. And here are the slides that I'm going to be using. Most of them are going to be coming from the Ohio State University Medical Center. And for the hepatic for the portal triad, I will be using a slide from the Practice Anatomy Lab from Mastering A&P. So here I am at the Ohio State University Medical Center, and I have clicked on duodenum. And so here's a very low magnification here. And remember that there are three structures that increase surface area in the small intestine. The first is called the placentae circulares, or sometimes called circular folds, and those are the permanent folds of the mucosa and submucosa. And that's this really large indent here into the lumen, and this would be a large indent into the lumen, so that would be another placentae circulares. Then as I get closer, I can see that on this big fold I see some smaller projections. These are the villi. And then as I look at the surface of each villi, I have this is my mucosa, and so I have simple columnar cells here. And at the surface of all of these, you can kind of see as this fuzzy area here at the end, these are going to be the microvilli. So those three structures, the placentae circulares, the villi, and the microvilli, all increase surface area for absorption. Since this is one villus right here, I should have a lacteal in the middle, and it's kind of this white area at the center of these villi. You'll see a lacteal. And as I go here, so let's talk about the layers here real quick. I'll zoom out again. So all of this kind of dark purple is your mucosa. Then this area is the submucosa. Here I have the muscularis externa, and then I'm going to have the serosa as the very last outer layer. And in this submucosa here, these are the intestinal glands, and they're going to be producing mucus and some other secretions that will be going out into the lumen. Then as I go down, I have my 2 layers of my muscularis externa. This first layer is the inner circular layer, and then this thinner portion here is the outer circular layer. And then this thin portion again is your serosa. Alright. Just to give you an idea here, this is the pancreas. Underneath, we got a slide of that. Okay. For the colon, or the large intestine, this slide looks kind of upside down. This purple is the mucosa, this is the submucosa, and a larger muscularis externa, although the only thing we should see here is the inner circular, because the outer longitudinal layer has narrowed down to that one little band called the tinea coli, and then there would be a serosa around the outside. But what I'm looking for are the goblet cells, and so we're going to see a lot of them. All of these white kind of circular globs here are goblet cells. So you can see that we have them scattered throughout the entire large intestine here. For the liver, this is the first accessory organ of the lower digestive tract. And remember I said that the cells are located or arranged in this lobule. You can kind of see in this stain here how they're in lobules here. And at the center of each lobule, I said there was a central vein. So kind of this hole or this circle in the center here is the central vein. And leading to that central vein are all of these little white canals. And those white canals are called sinusoids. And making up those sinusoids on each side, I can see some individual cells, and these are the hepatocytes on each side, kind of making this channel that's leading to the central vein. And at the corner of each lobule, I should have a portal triad, which had 3 different things. And I'm going to go to the practice anatomy slide to show you the different structures for the portal triad. Okay, in this picture, we see here are the hepatocytes again. The little white channels are going to be the sinusoids, and they're actually, from this one, they would be going in kind of away from this area. This is our triad. And so I have 3 things. I have a bile duct. And you can note they should be simple cuboidal cells. I have the arteriole, or a branch from the hepatic artery, bringing fresh blood from the heart. And then this large structure here, kind of flatter, is the branch from the hepatic portal vein. And this is bringing the nutrients from the digestive organs to the liver. So those are all within a portal triad at the corner of, at each corner of the liver lobules. And our last structure here is the pancreas. And remember I said pancreas is a mixed gland, both exocrine and endocrine functions. And it's going to look similar to the salivary glands that we saw in another presentation, because the exocrine portion is going to be made up of these serous acini, which will be very much like the serous acini of the parotid gland or the submandibular gland. But what distinguishes this from the submandibular, which was the mixed gland, is that I'm looking for these islands or pancreatic islets of cells, and this is the endocrine portion here. So I've got the exocrine portion, which is my pancreatic acini, and then my endocrine portion, which is this island or pancreatic islet. And so that's what I'm looking for to distinguish this as the pancreas as opposed to the salivary glands. Again, here's another islet, and these are the pancreatic acini. So I hope this video helps you identify some of the histology of the lower digestive tract.